Hello and welcome back to Biochemistry for Health Sciences channel. In today's video, we shall talk about regulation of metabolism by sequestering enzymes within specific compartments. As we discussed in the previous video, metabolism can be controlled by regulating the amount of blood that reaches the cell, since a lot of oxygen as well as nutrients travel through the bloodstream to every part of the body. Since metabolism requires the transport of various substrates, intermediates, and/or products across membranes, another way to regulate metabolism is by regulating transport proteins. And since metabolism involves a lot of biochemical reactions, and these reactions are catalyzed by enzymes, it's not surprising that probably one of the best ways to regulate metabolism is by controlling the enzyme function as well as the enzyme number. There are several ways to change or regulate the catalytic activity of existing enzymes within the cell. Today we'll focus on sequestering enzymes within compartments. Cells in our body can be as simple as a red blood cell, which basically has the plasma membrane and therefore only one compartment. However, most cells in our body have multiple compartments. So not only do they have the cytoplasm that is surrounded by the plasma membrane, they also have many other membranes, such as the mitochondrial membrane that forms the mitochondria, the nuclear membrane that form the nucleus, and so on. Recall that some metabolic pathways occur in specific compartments or specific organelles. For example, glycolysis occurs in the cytoplasm, PDH, TCA, the electron transport chain oxidative phosphorylation in the mitochondria, glycogen metabolism in the cytoplasm, HMS in the cytoplasm, triglyceride synthesis in the endoplasmic reticulum, synthesis of phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, all in ER, cardiolipin synthesis in the mitochondria, ketone body synthesis in mitochondria, and cellular digestion, which occurs in the organelle that we call lysosomes. And then there are certain metabolic pathways that occur in two or more specific organelles. Examples are gluconeogenesis, where the pyruvate oxaloacetate reaction occurs in the mitochondria. The rest of the pathway occurs in the cytoplasm. Fatty acid synthesis occurs in the cytoplasm, while elongation occurs in the ER as well as the mitochondria. Fatty acid breakdown occurs in the cytoplasm as well as the mitochondria and peroxisomes. Cholesterol synthesis in the cytoplasm as well as the ER. Plasmalogens are synthesized in the ER as well as peroxisome. Ceramides are made in the ER and then converted to sphingolipids in the Golgi membrane. Most of the nucleotide metabolism occurs in the cytoplasm, a little bit in the mitochondria. DNA synthesis as well as RNA processing, that occurs in the nucleus for the nuclear DNA as well as the mitochondria for the mitochondrial DNA. Protein synthesis involves the cytoplasm, ER, Golgi, as well as the mitochondria. So since the metabolic pathways occur in specific organelles, it's very important that the enzymes involved in the metabolic pathway or any transport protein that is involved in the metabolic pathway must be transported to the right location in the cell. So for example, these enzymes of the Krebs cycle, the TCA cycle, although they are made in the cytoplasm, they must be transported to the mitochondria in order for the TCA cycle to function. Any defect in the transport of the enzyme as well as the transport protein that is involved in the metabolic pathway may eventually lead to a disease. In several metabolic pathways, there is a transport of the substrate or one or more intermediates or other products across the compartment membrane. And in this case, this transport across the membrane allows yet another step for regulating the metabolic pathway. For example, the protein that carries citrate across the mitochondrial membrane, what we call the citrate carrier or CIC, this protein is inhibited by certain polyunsaturated fatty acids. It's also inhibited by starvation conditions and is activated by thyroid hormones. So this inhibition or activation of CIC can be used to regulate metabolism either in the mitochondria as well as the cytoplasm. So by having compartments, it makes it much easier for the cell to regulate enzymes. If all of these enzymes were in one compartment, it would be very difficult to regulate all of these enzymes at the same time. But by having them in compartments, for example, the Krebs cycle is in the mitochondria, it's much easier to regulate the Krebs cycle by regulating the enzymes in the mitochondria. Also, by having compartments, that allows another way to regulate metabolism, and that is by regulating the transport of either the substrate or one or more intermediates or the product across the compartment membrane. So this is one way by which cells can regulate metabolism, and that is by sequestering enzymes in specific compartments. In the next video, we shall look at the second way of regulating metabolism, of regulating enzyme, and that is the availability of cofactors. So until the next video, thank you very much for watching. Goodbye and have a wonderful day.